In my talk, I'd like to share with you our research efforts on the development of a novel therapeutic strategy targeting the replication machinery and pathogenesis of SFTS virus. Severe fever with thrombocytopenia syndrome, SFTS, is a serious tick-mediated viral hemorrhagic fever caused by SFTS virus identified in China in 2011. In 2013, the first domestic case was reported in Japan. As of May 2020, mostly from western part of Japan, more than 500 cases have been reported. The fatality is between 10 to 30%, which is high, and it is caused by Hemaphysalis longicornis, widely found in Japan. In Japan, there's a report of infection from China from human to human. In Japan, there's a report of human infection caused by a pet developing SFTS. So it is important emerging viral infection from public health point of view. Appropriate prognosis and therapy need to be promoted. However, currently, antiviral drug favipiravir has a clinical study underway, but there's no approved effective therapy or antiviral drug nor vaccination available. First, let me talk about the classification of SFTS virus. In Phenylviridae family, SFTS belongs to genus Bandavirus, and there's also genus Ook virus. In these, there are many viruses that are mediated by ticks, and mostly they are infecting warm-blooded animals, so it is classified as tick-mediated phenylvirus. And among these viruses, in genus Ook virus, there is no report which causes pathogenesis to humans. But in Bandavirus, we have SFTS virus. There's also heartland virus causes serious hemorrhagic fever. There's also Banjavirus, which causes febrile disease accompanying with the CNS symptoms. So there are viruses pathogenic to humans in the virus group of Banda. Let me now talk about the SFTS virus, which causes SFTS. It forms envelope viral particle, which is round or in polyform, and it has, as a genome, 3 segmented negative RNA. There are segments L, S, and M, and the non-coding region of genomic terminal functions as promoter to control viral gene expression and replication. Segment L has L protein RNA polymerase for viral RNA replication and transcription. Segment M has glycoprotein GN and GC coded for viral absorption and penetration to cell. Segment S encodes protein N that forms nucleocapsid as an ambisense genome, and NSS, non-structural protein that regulates virulence. Now I'm going to briefly talk about pathophysiology and treatment. SFTS is a serious systemic febrile disease with GI symptoms, neurological symptoms, thrombocytopenia, and leukocytopenia. In SFTS, virus proliferates in immune cells in the secondary lymph node organ. The spread of further infection induces systemic inflammatory response syndrome or SIRS with cytokine storm. In severe patients in the blood, high level of inflammatory cytokines and chemokines are detected. Enhanced systemic inflammatory response triggers, in severe cases, fatal conditions including hemophagocytic syndrome, disseminated intravascular coagulation syndrome or DIC, bleeding tendency, and multi-organ failure. With regards to the treatment, in the early to mid-term of infection, if possible, antiviral therapy by favipiravir is implemented, and for serious conditions, we need to control excessive inflammatory response, we need to control DIC, and symptomatic therapy is needed. But if you have cytokine storm, DIC, or hemophagocytic syndromes and multi-organ failures, with the progression of disease, treatment is difficult. That leads to high fatality. In severe cases, in the blood, high level of inflammatory cytokines and chemokines are detected. So the development of cytokine storm are suggested to be related to the prognosis and severity of the disease. So we looked at the infection of SSTSV, whether or not that will induce inflammatory response. We infected human monocyte-derived cell, which is one of the infection-targeted cells, with SFTSV and heartland virus that causes SFTS-like disease in human, and analyzed the proliferation and production of IL-8, which is inflammatory chemokine detected in severe human disease. Both viruses in human monocyte proliferated efficiently, and high level of IL-8 production was sustained for a long time. This result suggests that SFTS virus infection and proliferation in the target cell induces high level of sustained inflammatory response that leads to the systemic response. For effective therapy of SFTS, the useful strategy is to explore and develop medicine that will target and inhibit simultaneously the SFTS viral proliferation, uncontrolled increase of inflammatory response caused by viral infection, and the amplification of systemic inflammatory response caused by the cytokine storm. In the body, inflammatory response is induced through a variety of pathways, but in this talk, we will focus on NF-kappa B, a representative for inflammation induction pathway. Infection, stress, and inflammatory signal through several signal transmission pathways activate NF-kappa B transcription factor complex. Following nuclear translocation of the transcription factor complex, inflammatory mediators, including cytokines, are expressed. It is suggested that SFTS virus induced inflammatory response through NF-kappa B pathway. Earlier, I mentioned that NSS non-structural protein of SFTSV is virulency regulating factor that controls the host immune response. The studies so far show that NSS has a function to inhibit interferon response, which is an important antiviral response of the host. It is suggested that this NSS function is important for efficient viral proliferation in the infected cells and in the host. On the other hand, it is suggested that NSS, by activating NF-kappa B pathway, can induce inflammatory response. So, we express NSS protein from SFTSV and that of bandavirus on human-derived cell and study the NF-kappa B signal transduction pathway activation capacity and comparison. Both kappa B promoter luciferase plasmid that induces the luciferase expression by activated NF-kappa B transcription complex and NSS expressing plasmid were introduced in the cell. Luciferase activity expressed by NF-kappa B was used to measure NF-kappa B pathway activation capacity or inflammatory response induction capacity. Viruses in genus Bandavirus are known to have different pathogenicities to humans. Highly pathogenic SFTS virus and heartland viruses cause severe hemorrhagic fever-like disease to humans, while Bandavirus causes febrile disease accompanied with neurological symptoms to humans. human pathogenicity of recently discovered novel virus close to SFTSV is unknown. So we analyzed and compared NF-kappa B activation by NSS of viruses with varying pathogenicity to human, including SFTS virus, heartland virus, and Banja virus, and newly identified Hunter Island virus, whose pathogenicity to humans is unknown. Y-axis is luciferase activity, x-axis shows NSS of various viruses. From this study, we can see only cells with highly pathogenic NSS expression that cause SFTS or SFTS-like disease induce high activation of NF-kappa B pathway. This interesting result suggests that induction of inflammatory response through NF-kappa B pathway by NSS of SSTSB plays a role in the induction of mechanism of SIRS in SFTS. Thus, proliferation of SSTS virus in the target cell induces the production of inflammatory cytokines and chemokine, and NSS that regulates virulence activates NF-kappa B pathway. So we conducted a study to explore antiviral compound that inhibits signal transduction pathway and activation of NF-kappa B as well as SFTSB proliferation. For the exploration and development of antiviral drug, antiviral activities of many drugs and compounds need to be tested and evaluated. Normally, antiviral activity of a drug is evaluated with infectious viruses, but experiments using SFTS virus require biosafety level 3 facility, making it difficult, therefore, to develop antiviral drug of SFTS. So we try to establish a model system to reproduce part of infectious cycle without using infectious virus. By using this system, we can carry out high-throughput screening of antiviral drug and medicine in the ordinary lab safely and efficiently. This shows the proliferation cycle of SFTS virus. Like many of the viruses in order Bunyaviridae, proliferation cycle of SFTS virus includes steps such as attachment and infiltration to cell, membrane fusion, uncoating, RNA transcription and replication, particle assembly, embedding, and extracellular release of progeny viruses. In this cycle, steps such as viral messenger RNA transcription by RNP complex, protein expression, viral genome replication, and RNP complex synthesis play central role in replicating progeny viral particles. So they are ideal targets for antiviral drug development. So we try to structure mini-genome system to establish a model for RNA transcription and replication processes of SFTS virus. Mini-genome system is an experimental system in which, without using infectious virus, viral RNA synthesis and replication steps are reproduced by constituting RNP complex in the cell by providing miniature viral genome like RNA and viral protein essential for viral RNA transcription and replication from plasmid cDNA clones. First, plasmid for mammal cell expression is established, and that expresses proteins L and N, which constitute RNP complex. When these two proteins form RNP complex with viral RNA in the cell, viral RNA transcription and replication as well as protein expression processes can be reproduced. Next, in order to establish mini-genome system that imitates and segment non-coding region of a genomic terminal, including promoted sequence of viral gene is maintained, and gene that encodes viral glycoprotein is substituted with reporter gene, which is luciferase gene. Thus, mini-genome clones was established. cDNA that codes mini-genome is cloned in the downstream of human RNA polymerase I promoter and transcribed and supplied as mini-genome RNA in the cell. By introducing these three plasmid clones into human-derived cell, proteins L and N, which were expressed and supplied in the cell, and mini-genome reconstitutes RNP complex, thereby reproducing steps involving viral RNA synthesis and protein expression without infectious virus. Mini-genome replication and transcription by viral RNP complex can be monitored by measuring luciferase activity expressed from mini-genome. And this graph shows the activity of luciferase driven by mini-genome. Namely, this shows the mini-genome activity expressed by RNP complex of SFTS virus. X-axis shows plasmid with or without protein L expression. Y-axis shows luciferase activity expressed from mini-genome. Without the introduction of plasmid with L protein expressed in the cell, the luciferase activity is reduced by 1/1000th. So it was confirmed that this luciferase activity expressed from this mini-genome is dependent on the function of viral RNP complex constituted in the cell. Now that we have established a system that models the viral transcription and replication without using infectious SFTS virus, using this mini-genome system, we studied whether a drug that blocks NF-kappa B signal pathway can inhibit RNA transcription and replication of SFTS virus. This time, we tested the mini-genome inhibition capacity of the 6 NF-kappa B signal transduction inhibitors. We then chose the one with high mini-genome inhibitory activity and evaluated its viral proliferation inhibitory activity by using infectious SFTS virus. We used favipiravir with its confirmed anti-SFTS viral activity as positive control. As a result of mini-genome screening, among the 6 compounds, only SC75741 showed inhibitory activity against mini-genome activity. X-axis is the drug concentration, Y-axis in red is the luciferase activity expressed from mini-genome. Black line shows cell viability. IC50 of SC75741 was 0.66 micromolar. This inhibitor is known to suppress inflammatory response by inhibiting the binding of P65 subunit of NF-kappa B complex to DNA. Also, its inhibitory effect on the proliferation of highly pathogenic influenza virus has been known. Next, we evaluated SC75741's inhibitory activity of viral replication. The drug administration was started following the viral absorption and measured the viral load in the culture supernatants 72 hours after infection and compared it against those without drug administration to evaluate antiviral activity of SC75741. The left graph shows the inhibitory activity of SC75741. The right shows the inhibitory activity of the positive control favipiravir. Y-axis shows percent viral titer reduction under infection. X-axis shows drug concentration. SC75741's IC50 was 0.2 micromolar and IC90 was 2.2 micromolar, showing strong antiviral activity against infectious virus. This antiviral activity was comparable to the anti-SFTS-free activity of favipiravir. In the first part of the talk, we showed that NSS of SFTS virus and heartland virus activated NF-kappa B signal transduction pathway. Since this NF-kappa B pathway activation by NSS is suggested to be the mechanism of the inflammatory response induction in the infected cells by SFTS virus and heartland virus, we then studied whether SC75741 can suppress NF-kappa B pathway activation induced by NSS from SSTS virus and heartland virus. Similarly with the previous experiment, SC75741 was introduced to the cell with NSS-expressing plasmid and kappa B promoter luciferase plasmid and measured luciferase activity. The 2.5 micromolar is the value of IC90 obtained in the previous study on viral proliferation inhibition. As shown on the x-axis, 2.5 micromolar of SC75741 was administered to NSS-expressing cell to test NF-kappa B activation suppression by SC75741. Compared with those without the drug administration and other NF-kappa B inhibitor, SC75741 inhibited the NF-kappa B pathway activation by NSS of SFTS3 to less than 50%. This study showed that SC75741 suppressed the activation, namely the inflammatory response induction of NF-kappa B pathway caused by the NSS expression of SFTS virus and heartland virus. Next, we study the inhibition by SC75741 of inflammatory response induced by inflammatory cytokines. In patients with SIRS, inflammatory cytokines produced by viral infection further induces the inflammatory response of non-infected cells. We studied whether SC75741 can inhibit the inflammatory response of the cells stimulated by inflammatory cytokines, namely TNF-alpha or IL-1 beta. Cells introduced with NF-kappa B promoter luciferase plasmid were stimulated either TNF-alpha or IL-1 beta, and the cells were treated with SC75741 or favipiravir positive control or with a control without drug, and measured luciferase activity expressed by NF-kappa B promoter. The result confirmed that SC75741 inhibits NF-kappa B pathway activation by TNF-alpha or IL-1 beta. On the other hand, favipiravir, that only targets viral proliferation, did not show inhibitory effect of the activity of NF-kappa B pathway stimulated by TNF-alpha or IL-1 beta. This suggests that SC75741 can suppress induction and amplification of inflammatory response stimulated by inflammatory cytokines. This is the summary up to this point. In our research, we showed that ST75741, an NF-kappa B signaling inhibitor, inhibits viral RNA synthesis of SFTFS mini-genome and further inhibits the proliferation of infectious virus. Since ST75741 inhibited activation induction by NS of SFTS virus of NF-kappa B signaling pathway and showed inhibitory effect of inflammatory response induction stimulated by inflammatory cytokines, the compound can be a drug candidate to suppress both the increased inflammatory response and viral proliferation caused by SFTS viral infection. In the future, we need to validate ST75741's effect on protection against viral infection as well as treatment effect using SFTS animal model. ST75741 is only an experimental drug. Among the candidate drugs to be clinically used in the future, drug candidates with similar effect need to be identified to develop the treatment by ST75741. The mini-genome system that we established was shown to be a useful means to safely and efficiently identify antiviral drug in the early stage of drug exploration without having to use infectious virus. Finally, based on the research results, I'd like to discuss the mechanism of proliferation and pathogenicity of SFTS virus. Among bandaviruses, only SFTS virus and heartland virus are known to cause hemorrhagic fever or SFTS to humans. In the research, we showed that NSS from SFTS virus and heartland virus compared to NSS of other bandaviruses have higher capacity to activate NF-kappa B pathway. This suggests that NSS is the determinant factor of the pathophysiology of the disease caused by bandavirus, regulating the pathogenicity. On the other hand, ST75741, an NF-kappa B inhibitor, inhibited viral proliferation and RNA synthesis by SFTS mini-genome. It suggests that interaction between viral RNP complex and NF-kappa B pathway is an important host factor for the SFTS viral proliferation. It seems that SFTS virus activates NF-kappa B signaling pathway through NS and thereby promotes viral proliferation. The biological significance of inflammatory response in the SFTS viral infection can be discussed as follows. In SFTS viral infection, NSS activate NF-kappa B signaling pathway to promote viral proliferation. The consequent induced and amplified inflammatory response by the host may play a role in the development of the SFTS pathophysiology. We plan to research proliferation and pathogenicity of SFTS virus and their relationship with the host inflammatory response. Last but not least, I'd like to thank all the collaborators of the research. The research that presented has been carried out in our lab by Crystal Mendoza as leader. Thank you for your kind attention.